You're listening to the Valley Current.
So are you fully vaccinated at this point?
I am. I got my second shot on Saturday.
So how do you feel?
Perfect. I had absolutely no side effects whatsoever.
Nice.
Other than what I had after the first shot, which was a area about as big as a Morgan silver dollar around the inoculation site that had tenderness when I pressed my finger on it. And that was it. That was exactly what happened this time as well.
So when you think back to all the shots you've gotten over your 90 plus years, was this the easiest one of all, or how do you feel about it?
Well, I can't really remember the ones I got as a child, but do you have a smallpox vaccination?
Yes. Yeah, I remember getting that.
You and I are in the age group where that exists, but there hasn't been a smallpox inoculation for 20 years in this country. Mostly because, and that's an easy vaccination because it's just a scratch with a needle.
Right.
It's not an injection.
Right.
But I have had no side effects at all.
Well, you know what? You are probably the poster child for Dr. Fauci to put up there and say, look, if Dr. Leonard Hayflick, who is going to wait, according to his own podcast, to the end of the year, accelerates by 6+ months, then it's time for everyone to accelerate by 6+ months or more and get their vaccinations, right? You're the poster child.
Well, I don't know, but Tony is not a spring chicken himself.
Right.
He's in his 70s or 80s, maybe.
Well, I'm sure he's aged over this last couple of years, for sure, last year.
That's for sure.
Right. So congratulations on winning the chance to speak to the group that studies cell senescence. There are very few people that even understand that word of cell senescence. And that's kind of the topic today, because it is a big topic, but I don't think many people understand that cells do die, and they don't necessarily, with few exceptions, you'll tell us about the exceptions, they don't replicate forever. And you have a limit, which is a limit you discovered called the Hayflick Limit, that I believe you will get the Nobel Prize, hopefully sometime before you hit 100. And your mom did last over 100, so I guess you have some room to run there, and you're in good health and you're taking care of yourself and you got your vaccination. So tell us about that word and when you first came about to even learn about cell senescence in your own career, Len, because this goes back to a youthful time for you, right?
So you want to know about senescent cells?
Yeah, just give us your sort of summary version of what's in that PowerPoint. I will put that PowerPoint up because I think you've done a great job of preparing. But yeah, in general, I was very impressed that you are living in what I would call the moment of your career when you could actually look back at your early picture, which was on that presentation. I'm guessing you're in your 30s. I'm going to put it up on the screen just so you see it.
I don't remember.
But you certainly still have the same look. I mean, it is definitely you. Of course, you've got a full black hair and you're looking you're looking quite youthful there.
That's the poster, right?
And there it is, the discovery of cellular senescence. That's the topic. That's the PowerPoint. You're the person who did it. You're, at that point, a professor of anatomy at UC San Francisco. But tell us more about what you're going to present on June 1, because we're actually getting sort of a look-see ahead of time at what you're doing here in this presentation.
Well, the discovery involves the use of cell culture, which means the ability to grow human and animal cells in laboratory glassware or now in more modern years, plasticware. And that has been done roughly since the early 1900s. And from that time until the 1960s, when I entered the field, it was believed, and in fact it was dogma, that when cells are put into culture, they can be subcultivated. That is, the cells are put into a culture vessel and they grow on the floor of the vessel in a nutrient medium, and they replicate until the floor is covered, and then they stop dividing. And then if you want more cells, it becomes necessary to remove the culture fluid and remove the cells from the floor of the flask, which you do with a specific enzyme. And then you can divide that population of cells, which numbers in the multiple millions, and plant half of it, let's say, for simplicity's sake, plant each half in two separate similar culture bottles, and the cells will replicate in both bottles, of course, on the floor. And then you repeat the process 5 or 6 days later, after the cultures have been incubated at body temperature in an incubator that are commercially available. And you can do that many, many times. And you can increase the number of cells that you can then obtain in enormous numbers. It was believed for years and years, because of technical failures on the part of people, that if cells did stop dividing in culture using the procedure that I described, it was because you were ignorant of the nutrients that they needed. And in fact, that area of the nutrient requirements of cultured cells, including human cells, was a huge area of interest on the part of dozens, if not hundreds, of scientists in the 40s, 50s, and 60s.
Right.
And whenever the cells stopped dividing, people attributed it to all sorts of excuses: failure to wash the glassware then in use properly, failure to know what nutrients were required, on and on and on, and other kinds of technical failures.
Right.
Well, I was in a position to challenge that belief because I had been taught to believe that as well. And without going into the details of why I started these experiments, they really had nothing to do with the phenomenon that I discovered or with the project that I had been interested in doing, because this observation was an accident, actually. What I was trying to do, and I'll say this very briefly, was to find out whether some human cancers were caused by viruses, which was known to happen in other animals, in chickens and rodents. And I thought, well, it's possible that some human tumors might be caused by viruses. And so I decided to do a very simple series of experiments in which I would grow cancer cells in culture and then extract the cells using the techniques that were used to make those virus discoveries in animal tissue, and expose the extracts to normal human cells. Well, obtaining cancer tissue, which I did obtain from the University of Pennsylvania Hospital across the street from the Wistar Institute, where I was working at the time, cancer tissue was very easy to obtain. It could be delivered almost daily from the operating rooms there. But the difficulty was to get normal human tissue, which normal humans don't sacrifice without serious complaints. In addition to which, I wanted to culture human cells from human fetuses, because it was known at that time that human cells from adults often contain unwanted viruses like herpes simplex that cause shingles, or measles virus, or other known viruses that would have interfered with my work. But if you use fetal tissue, it was known that the fetal tissue was free of such contaminating viruses. And so I chose to get fetal tissue.
Well, that was a bold move, and got the Vatican all upset with you, didn't it? I mean, just to dovetail in some other remarks that we've made on these podcasts, because I put up this slide of where you really start off challenging what a Nobel Prize winner in 1912 fundamentally had wrong, right? Isn't that your point?
I didn't start off to challenge anybody because what I found was an accident. I had no interest in aging at that time. I was only interested in cancer research,
Right.
as I explained. So what came later, it turned out to, in fact, torpedo Alexis Carrel's belief.
But that's the big part of science that gets unappreciated, which is critical thought and also just luck or accidental findings, kind of like what happened to you, right?
Yeah, many major findings in science are accidents of observation or circumstances. So to finish the story, finally was able to get fetal tissues sent to me at odd times over a period of many months because it was unpredictable when an abortion, either spontaneous or surgical, would be available. After I had arranged with the University of Pennsylvania Hospital to get such tissue, I cultured the cells from the lungs of the fetal donors because that was a large organ at the time that they were sent to me, and provided a lot of cells that I could use in order to determine whether they would be susceptible to a putative cancer virus. And in the course of doing that, I subcultured the human fibroblast, which is the name of the cell that grew best from the lung tissue. They were subcultivated exactly as I explained at the outset of this discussion. And after I had received over a period of about 10 months, a dozen or more fetal tissues that I had cultured, and each 4 or 5 days, they all underwent subcultivation. I found that some cells in one culture were not dividing, and that was a little troublesome, although at that time it was very common for that event to happen because of contamination with microorganisms that grow well in that tissue culture media, or some technical failure that I was unaware of. So I just ignored that single event. And as the weeks went by, other cultures and a dozen or so that I had growing from a dozen separate fetuses, a couple more were showing the same phenomenon. That is, cells stopped dividing, despite the fact that I was using the same media for all of them, the same stack of glassware that was used for all of them, and the same technician was culturing them. So that was an anomaly. And then I decided to look at my laboratory notebooks. I found that the cultures that were in which the cells had stopped dividing were obtained from the embryos that were received farthest back in time. It was a consistent observation, despite the fact that they're all growing under the same circumstances. Only the oldest ones stopped dividing.
Right.
And that was not the eureka moment. It was the that's funny moment.
Like curiosity of observation that something's going on here, right?
Exactly. And so I began to look into that question of why these cells only went through about 40 to 50 doublings of their population and then stopped dividing.
Right.
In addition to which, at that time, the technology was known for freezing cells in liquid nitrogen or solid CO2 freezer, or even electrically driven freezers. At very low temperatures, you could freeze the cells, and they would, of course, be incapable of dividing, but they would remain viable or alive under those conditions.
Right.
What I discovered that if you made ampules of cells from, let's say, the first doubling all the way up to 40 or 50, and then you reconstituted an ampule from the cells, let's say, just arbitrarily at the 20th doubling, the cells remembered that they were at the doubling level of 20 when they were frozen, and months, and now even 50 years later, They retained the memory of the population doubling level that they were at when frozen. And when thawed, they increased doublings up to the maximum of 50. So the cells have a memory. And I began to look at that question because that was a very unusual observation that cells could remember where they were in the doubling events, and did some experiments with my then student, Woodring Wright, and we discovered that the replicometer, which I called this imaginary counting mechanism, it had to be a counting mechanism.
Right. It's almost like a computer inside a cell.
Yes, it was located in the nucleus of the cell.
Mm-hmm.
Now, I had in scientific papers, when you make an observation like this or anything else, you are given the opportunity in the paper to put in a section of the paper called discussion what your interpretation of the events are that you have found. And I could not find a reasonable explanation for what I had found, other than the possibility that this was telling me initially about aging or senescence. That is, cells stop dividing at some particular point during the aging process. And I invoked that explanation as a suggestion. There is no evidence for it, of course, because there are no accepted markers for aging even to this day that are universally accepted.
Right.
So this discovery of senescence, as I called it, remained in the scientific literature for many years. Most people in the field didn't believe it. They thought I had made a mistake in my culturing techniques, although I did the unpublished experiment of sending cultures to leading people in the field who I knew well, and told them, I'm sending you this culture today, and in 3.5 months, the cells will stop dividing.
Right.
And they said, oh, that's not possible. Sure as heck, 3.5 months from the time I sent them the cultures to them, my telephone rang from all of them saying the culture stopped dividing. There were other evidence that I had, and I won't go into the details now. It's all described in that PowerPoint presentation. But my observation was not believed for close to 10 years until discoveries in an entirely different field by accident came together. The entirely different field was the field of work that was done by two lady scientists, Elizabeth Blackburn and Carol Greider, who worked with a protozoan called Tetrahymena, and they discovered in those organisms that they had what are called telomeres. Telomeres are the ends of chromosomes, and they discovered that those telomeres, that up until that time, it was unknown what their role was, but it was discovered by them that the role was to protect the ends of chromosomes when the chromosomes divided. I won't go into the details of that. In addition to which, they also discovered an enzyme called telomerase, which they thought had something to do also with the replication of that organism. Then the critical accident happened. One of my students, I'll call him a student, technically he wasn't, but he was a close associate of mine who didn't work in my lab, but he was very interested in what I had found. His name was Calvin Harley. He was at that time on the faculty at McMaster University in Canada. And Carol Greider, who was working at that time, I think at the University of California in Berkeley, or else at Johns Hopkins, I don't remember which, But she went to see her boyfriend at McMaster University in Canada. And because she was a scientist, they invited her to sit in on their discussions that the faculty was having, including the discussions with Calvin Harley, where she learned about my system that I just described. And the two of them had the brilliant idea of trying to determine what happens to the telomeres in the cultures of the human cells that I had described. And they made the very significant discovery that was published ultimately, of course. They discovered that as the cells in my system replicate to the final point, the telomeres, the ends of the chromosomes, shrink in size and become smaller and smaller because the chemicals that construct the telomeres, part of those chemicals at the ends of the chromosomes fall off at each division until a critical short length is received that signals to the cell to stop dividing. And that was the molecular explanation for my phenomenological finding, in addition to which I made the very important suggestion based on my work that normal cells have a finite capacity to replicate, and cancer cells are immortal. They can replicate forever.
Right. It's almost like cancer, which is thought of as associated with death, has sort of the interesting property of defying cell senescence because these cells just wildly take off, right?
Well, what I discovered was that in contrast to normal cells that have a finite capacity to replicate, cancer cells have an infinite capacity to replicate. And the explanation for that was then discovered in the capacity of this enzyme called telomerase that gets expressed in cancer cells, but not in normal cells. The telomerase adds on to the ends of the diminishing chromosome ends the missing chemical pieces and therefore maintains the telomere length constant, thus providing the cancer cells with the property of immortality. And that has been confirmed, of course, many, many times. The two ladies, Carol Greider and Liz Blackburn, won the Nobel Prize in 2009, not for what I described that's related to my work or having anything to do with aging. The Nobel Prize was awarded to them because it was interpreted that telomeres protected chromosomes, and that was the reason they were given the Nobel Prize. It was not given to them for any association of what they found with aging. The reason I mentioned that is that even Liz Blackburn, initially when she learned about the results of Greider and Harley, denied that what she had found had anything to do with aging. Subsequently, a few years later, she embraced my interpretation of telomere shortening as telling us something about cell senescence.
Quite a series of random acts, if you think about it, because there's you making an observation that was against the grain of what the typical view was at the time, for which there had been a Nobel Prize winner for.
Well, that came 10 years later.
Yeah, I mean, no, but a Nobel Prize winner from 1912, the original view of.
Oh, yes. Well, right? I torpedoed that.
Right.
Varel did not get the Nobel Prize for his belief that cultured cells were all immortal. He got the Nobel Prize for his invention of a surgical knot.
But I mean, it's such an interesting series of events because it takes two women coming up with something else for your work to actually become acceptable. Like, that's an interesting aspect even of itself, right?
Well, that was a key reason for my work being accepted. But during the preceding 10 years, I had identified the location of the counting mechanism in the cell nucleus, which is where the telomeres reside and telomerase gets expressed. During that 10-year period, that my work was not believed, certainly not in my interpretation, which later, even to this day, is now believed, but it had been repeated by many people worldwide. So the phenomenon was known to be real, but the interpretation remained until more recent years. Even to this day, there is essentially a huge industry that has developed in the past, I would say, 8 to 10 years in this country and in many other countries, in which new companies have been formed to study this phenomenon of senescent cells, because it has now been found that the senescent cells that I found in laboratory bottles are now found in humans as we age. Those are called senescent cells also. Then people began to think that, well, maybe there is a way to get rid of those old cells in old people. And the term that was invented to describe that was senolytics. And if you look it up now on a search engine, you will find maybe 100 or more companies that have been founded by investors looking for ways to destroy or inactivate senile cells in humans and in animals. So my observation has now received that kind of attention, in addition to which the attention that I didn't know about until about 6 weeks ago, that I described in the message that I sent to you and other friends from this Society for the Study of Senescence, located in the United Kingdom, Durham University, I think, that I had no knowledge about whatsoever, although it was in existence for a couple of years. Hundreds of members, and those are the folks who invited me to give a presentation on the history of my discoveries next week on June 1st.
Right. I mean, that's a very amazing thing, because this idea of cell senescence, you would say, didn't really promote startup businesses until relatively recently. And I don't know what instigated that. You could say, well, this whole view of people wanting to live to 120, or people believing they can live to 120. Obviously, our colleague and best good friend and my next door neighbor, Dr. Walter Bortz, firmly believes that you can live healthy 100 years. He's written about it. He's got a book called Dare to Be 100. He is also in his 90s. And, I think at the end of the day, there's like this new instigation of aging, and aging in a healthy way, that has become subject to the eyes of venture capitalists, which we haven't seen before. And they are promoting all sorts of biotech businesses that you could either argue are going to try to sell some snake oil, or maybe are going to come up with some science here that suggests ways to have a longer lifespan and a longer health span, right?
Well, that's their hope, but I'm not terribly optimistic about the likelihood of their succeeding, because my understanding of the aging process is such that it is not a problem in biology, it's a problem in physics. And it involves the second law of thermodynamics, which means that these startup companies, if they're hoping to achieve what they want to achieve, will have to violate a fundamental law of physics. What they're gaining, however, is the sale of stock and the income they get from the sale of stock, and the hype about their hopes for the future. But I'm very negative about what they think they're going to succeed in doing. This effort to interfere with the aging process has been going on ever since human history has existed, which means 3,500 years ago. And each generation believes that they have the knowledge to do it. We laugh, of course, about efforts to do this, even as recent as the 19th century. But during the 19th century, those scientists believed they had the knowledge to interfere with the aging process. And the same thing exists now in the 21st century. So that each century believes they have the way to do it. But I'm, again, not optimistic at all. And furthermore, I think that even if it was possible to do it, the events that might occur as a result of interfering with the human aging process would be enormous. And most of them would be negative in respect to our civilization.
Well, people want to believe that they can extend health span and lifespan and maybe even work span. And they want to believe that there are no consequences. Obviously, there are huge financial consequences. There are lots of people that will run out of money. Now, maybe the robots will exist in such a huge way that we'll have a leisure class society where work is irrelevant. But tell us a story about how you tried to get the paper published and you got these rejection letters, because it's such a classic example of science being unwilling to deal with the unfamiliar. And, I'm sure this is a very polite letter when you look at it, but I'm sure when you first read it, you were saying to yourself, hey, are you the Journal of Experimental Medicine, or what are you if you're not going to publish this paper?
Well, the history of it is also interesting, and that is that I decided to send the paper to be published to the Journal of Experimental Medicine, which you can see at the top of the letter of rejection. That journal was an old and established journal that went back many, many decades, and in fact, published all of the work of Alexis Carrel, who was the scientist who claimed to have subcultivated and carried chick heart fibroblasts for over 40 years. And on the basis of his work, he believed that all cells in culture had the potential to be immortal. But that was one reason I sent it to that journal. Another reason was that a more modern person by the name of Theodore Puck, who was a very well-known scientist at that time, also published in this journal, and he published his work with human cells, the same cells that I worked with. And I had learned by the time I submitted this for publication that he had missed discovering what I had discovered, because he believed that when his normal cells stopped dividing, as they did for me, he believed the old dogma that it was because he didn't know that much about their nutritional requirements. And I had done experiments that disproved that as a hypothesis. And so for those two reasons, I decided to send it to this journal. And they were very slow. It took about 5 or 6 months before they replied in the letter that you see, in which they rejected publishing my paper for the very reasons that were the major discoveries described in the paper. As you see, the editor, Peyton Rouse, said that I won't quote it exactly, but pretty close, as everyone in tissue culture knows, if you provide cells with the proper milieu in vitro, they will divide forever. Well, that was the very idea that we torpedoed. In addition to which, he said that my interpretation of what I had found as having something to do with aging was, quote, notably rash.
Ah. Ah.
So the notable rash turned out to be one of the major findings in the field of biology of human aging. But anyhow, he went on to win the Nobel Prize 3 years after he made that statement that you see on the letter.
Stay tuned for part 2 of this episode of The Valley Current.